Dear students, today I will discuss on hemagglutination assay and hemolysis assay. Hemagglutination is clumping of red blood cells, whereas hemolysis is lysis or destruction of red blood cells. I will discuss the principle and the procedure involved in both the assays. Now we know that agglutination or clumping takes place when specific antibodies react with particulate antigen. Particulate antigens include white blood cells, red blood cells, bacterial surface, etc. And the immunoglobulins which are involved in the agglutination process are called agglutinins, whereas the antigenic determinants which are present on the particulate antigen surface and are recognized by the agglutinins are called agglutinogens. And among the agglutinogens, red blood cells are very common. And when agglutination involves red blood cells, the agglutination is called hemagglutination. And the molecules which cause this hemagglutination, antibodies or viruses which are responsible for hemagglutination are called hemagglutinins. So hemagglutination can take place in the presence of antibodies or in presence of lectin proteins or also in the presence of hemagglutinins proteins which are present on viruses. Now hemagglutination takes place in the presence of antibody or immunoglobulins. It is a two-step reaction. In the first step, the immunoglobulin binds to the RBC, and at this step we can't see it with our naked eyes, but as the antibody RBC complexes cross-link together, which happens in case of multivalent immunoglobulins, the cross-linking forms an aggregate, large aggregate, and we can visualize that aggregate formation. Hemagglutination also takes place in the presence of some specific viruses like mumps virus, influenza virus, parainfluenza virus, measles virus. These viruses have a protein on their surface called hemagglutinin. This hemagglutinin protein recognizes specific molecules present on the RBC surface. They bind to the RBC surface and then cause hemagglutination. Hemagglutination also takes place in the presence of lectin proteins. Lectin proteins bind to carbohydrate moieties and they recognize the presence of specific carbohydrate moieties present on the RBC surface. They bind to it and then cross-link among themselves and form a large aggregate, or hemagglutination takes place. Hemagglutination assay is a method of quantification for viruses or bacteria which are capable of causing hemagglutination. And this assay method was developed by American virologist George Cabell-Hist in 1941. He discovered this hemagglutination assay and also anti-hemagglutination assay. Now I will tell you how we can perform this assay in our laboratory. The procedure varies with the nature of the virus or bacteria. Hemagglutination reactions take place in the presence of electrolytes. So we have to take specific buffer in all the wells. Then we will take the virus sample or bacterial sample and then we will dilute in a series of dilutions. We will make two-fold dilution starting from 1:2 to 1:1024 dilution. Then we will add fixed amount of red blood cell suspension in all of those wells and then we will observe the result. Now after addition of red blood cell suspension, we have to incubate it for 30 minutes at 4 degrees centigrade for virus and at room temperature or optimal temperature for bacteria. We have to incubate it at 4 degrees centigrade for virus because many viruses possess neuraminidase protein enzyme. This enzyme causes detachment of the RBC from the virus particle. For those viruses, we have to incubate at 4 degrees centigrade. When virus particles and RBC, they are at equivalent concentration, then only clumping and visible aggregate will be formed. But if the virus concentration is more, then individual virus will bind to individual RBC, no clumping or cross-linking will take place. At the same time, if virus concentration is very low or RBC concentration is very high, same phenomenon occurs and they will bind individually and no cross-linking will take place. So, in case of excess RBC, the RBCs, they roll down or slide down through the walls of the tube and they form a red button at the end of the tube. So, in case of a negative result, we see a red button, whereas at the equivalent zone, since their concentrations are equivalent, they will cross-link and form a red mat throughout the wall surface. These are the tubes where hemagglutination assay has been done. So, looking at the wells, we can say which one shows negative result and which well shows positive result. These are the wells where hemagglutination reaction has been taken place, and these are the wells where RBC concentration was more and red button has been formed at the bottom of the tube. Hemagglutination is the basis of blood group compatibility. Individuals having blood group A has RBC with A agglutinogen. Individuals having blood group B have RBC with B agglutinogens. Individuals with AB blood group have RBC with both A and B agglutinogens. And in case of blood group O, neither A nor B agglutinogen is present. And in case of blood group A, anti-B antiserum is present in the serum. In case of group B, anti-A antiserum is present. So, if an individual having blood group A accepts blood from blood group B, what will happen? The anti-A present in the serum of blood group B will cause hemagglutination or agglutination of the RBCs. Hence, blood group A must take blood from another individual having blood group A, or he or she can take blood from blood group O. Hemagglutination is represented as hemagglutination unit. One hemagglutination unit is the minimum amount of virus required to form complete agglutination of the RBC. Suppose we have taken 25 microliter of virus sample and we have diluted it manifold, and suppose we observe complete hemagglutination in case of 1:64 dilution. That means 1:64 dilution contains one hemagglutination unit, and the titer value will be reciprocal of 1:64, that means 64 or 2 to the power 6. So, we can say 2 to the power 6 hemagglutination units are present in 25 microliter of the virus sample. The major disadvantage of this assay is that this assay does not discriminate between the alive and dead virus particles. Can you tell me why?
Ma'am, as this reaction is between an agglutinogen and an agglutinin, the survivability of the virus is immaterial. Hence, this reaction cannot distinguish between a live virus and a dead virus.
Very good. Both live as well as dead viruses will show same level of agglutination or hemagglutination. Hemolysis is complete destruction of red blood cells, and due to the destruction of red blood cells, the heme is released, and due to the presence of heme, the blood or plasma looks cherry red or dark red. And since no cell is present here, after hemolysis the serum is transparent. Now, hemolysis inside our system can be caused in the presence of many gram-positive bacteria like Streptococcus, Enterococcus, Staphylococcus, and also in case of malaria, due to the presence of Plasmodium parasite, hemolysis can take place. Also, hemolysis is observed in many autoimmune disorders as well as in some genetic disorders like sickle cell anemia, glucose 6-phosphate dehydrogenase deficiency, etc. Hemolysis assay is very important because this assay can tell us which bacteria is able to cause hemolysis, which one is not. Many strains of the genus Streptococcus can cause hemolysis. Streptococcus is a genus of gram-positive bacteria and they normally divide along axis, thus they are found in chains. But most of the Streptococcus species, they are non-pathogenic and some are found on our system, in our mouth, skin. They are commensal bacteria for us. Rebecca Lansfield classified Streptococci into several groups A to O depending on the presence of carbohydrate antigens on their cell wall, cell membrane. Some species of Streptococcus, they are pathogenic to us. Okay, and they can cause hemolysis. Now, hemolysis can be of three types: alpha hemolysis, beta hemolysis, and gamma hemolysis. And they are differentiated depending on the differential characteristics and varying degree of hemolysis. Now, we perform hemolysis assay on blood agar plate or blood agar medium. Blood agar medium is an enriched medium. It is used for fastidious organisms and for the hemolytic organisms. It is used to differentiate among the hemolytic microorganisms. And blood agar medium is prepared by addition of 5% defibrinated blood to the blood agar base. And blood agar base is prepared by mixing pancreatic digest of casein, papain digest of soy meal, NaCl and agar. Blood is added to the sterile blood agar base, mixed properly and then poured on the Petri plate. Now, after solidification, what we do, we streak on the plate and then incubate it at 37 degrees centigrade. Now, in case of Streptococcus mutans, Streptococcus mitis, Streptococcus salivarius, after the growth of the organism, the colonies look dark and greenish. Okay, what happens here? The microorganisms, they produce H2O2 and this H2O2 oxidizes hemoglobin. In hemoglobin, the iron is in its +2 state. The +2 iron is oxidized to +3. Now, this state of hemoglobin is called methemoglobin, which has a greenish look. That's why in case of alpha hemolysis, the colonies become greenish. But the RBCs are not destroyed. Only the hemoglobin is converted to methemoglobin. So, this is called partial hemolysis, not complete hemolysis, and sometimes it is called green hemolysis. Now, in case of Streptococcus pyogens, Streptococcus agalactiae, complete destruction of red blood cells occur. The microorganisms, they produce an exotoxin called streptolysin. This streptolysin react with the cholesterol present in the cell membrane of the RBC and on many other immune cells, and then they form pores or holes on the surface of the immune cells RBC surface. Thus, they lyse the RBC. So, complete destruction of RBC takes place in the presence of streptolysin. We call it beta-hemolysis. Now, streptolysin is of two types, streptolysin O and streptolysin S. Streptolysin O is oxygen sensitive. That means its activity is reduced in the presence of oxygen, whereas streptolysin S is oxygen stable. Most of the strains of Streptococcus pyogens produce streptolysin S, okay, and which is oxygen stable, and they destroy the red blood cells completely, and white halo is observed surrounding the colonies. But a few strains of Streptococcus pyogens, they produce streptolysin O, which is oxygen sensitive. If we want to see beta hemolysis, in case of those strains, then we have to make an anaerobic pocket, and it is done by stabbing the inoculating loop on the agar plate. So, these are the beta hemolysis pattern of those Streptococcus pyogens which produce streptolysin O. Now, another example of beta hemolysis is seen in case of Streptococcus agalactiae and Listeria monocytogenes. Okay. They also produce a streptolysin which is weakly produced or which has lower activity. So, hemolysis is only seen under the colony. So, if we want to see beta hemolysis in this case, we have to remove the colony with an inoculating loop, and we can see hemolysis of RBC beneath the colony. So, this is also beta hemolysis, but the hemolysin is not active here. And the third group is called gamma-hemolytic streptococci or gamma-hemolysis, where no hemolysis actually takes place. The main example of gamma streptococcus is called Enterococcus faecalis, previously called group D streptococcus. They cause no hemolysis at all, and they don't cause any RBC destruction, and thus no change in color is observed in case of gamma hemolysis. Now, hemolysis assay is very important since it is used to detect the presence of hemolysis causing microorganisms. Now, before making the blood agar plate, we have to be very careful. The temperature of agar and blood before mixing is very important.
Ma'am, may I know why the temperature is most important thing in making the blood agar medium?
If the agar is too hot, then when we'll mix the blood with it, the RBCs may lyse due to the high temperature. And if the temperature of the agar is too low, then the agar may solidify before the addition of the blood. So, temperature of the agar is very important. At the same time, temperature of the blood is also very important. It must be maintained at room temperature. Cold blood may solidify the blood agar base, and we can't keep the temperature high because the RBCs will be destroyed. Thus, the temperature of the blood and agar must be maintained around room temperature. To conclude, today we discussed on the principle and procedure of hemagglutination assay and hemolysis assay. Hemagglutination assay is very important because it gives us an idea about the hemagglutination causing agents if present in a sample. It may be an antibody or virus particle, sometimes bacterial cell, etc. And we also discussed on how we can calculate the number of hemagglutination unit. Also, disadvantage of hemagglutination assay. The assay tells us about the hemagglutination causing agents, but it does not tell us about the number of alive viruses and dead viruses present in the sample, because both alive as well as dead give same amount of hemagglutination reaction. We also discussed on hemolysis assay. Many microorganisms can cause hemolysis or destruction and or lysis of red blood cells or erythrocytes. Many species of Streptococcus genus are hemolytic. Okay, and we can divide them into three groups: alpha hemolytic group, beta hemolytic group, and gamma hemolytic group. And these three groups differ in the mechanism of hemolysis, also degree of hemolysis. In case of alpha hemolysis, partial hemolysis takes place where the hemoglobin is converted to methemoglobin. In case of beta hemolysis, complete destruction of RBC takes place. That is why the zones appear as yellow. And in case of gamma hemolysis, no hemolysis actually takes place. So, we learned how we can perform the hemolysis assay and what is the importance of this assay. This assay tells us about the presence of hemolytic microorganism present in a sample.